Today, we're going to talk about FTIR, or infrared microscopy, a chemical analysis technique that combines FTIR spectroscopy with a light microscope. In this video, we're going to cover all the basics of FTIR microscopy, from how the FTIR microscope works to what it's capable of. Before we dive into what FTIR microscopy is, we need to have a basic understanding of FTIR spectroscopy first. FTIR spectroscopy uses infrared light to identify chemical species. We shine the IR light on our sample, and the IR light interacts with the molecules or compounds in that sample. This interaction is detected by an FTIR spectrometer, and then that information is used to create an FTIR spectrum. Every chemical species interacts with IR light in a unique way, so each FTIR spectrum that results is unique. That makes the FTIR spectrum kind of like a fingerprint for each chemical species. Just like fingerprints can be a powerful tool for identifying people, the FTIR spectra are a powerful tool for identifying molecules and compounds. Now, on a more practical level, there's three basic ways we can do these FTIR measurements: transmission, reflection, and ATR. These are called measurement techniques, and each one is useful for different kinds of samples. For transmission, the IR light shines straight through the sample before being detected. This technique is useful for very thin samples, like tissues or films. With reflection, the IR light bounces off the surface of the sample prior to detection, making this technique the go-to for analyzing reflective bulk samples, like a silicon wafer, or analyzing samples that are on top of reflective surfaces, like thin films on metals. ATR uses a crystal, which is usually made of germanium. The crystal touches the sample, and the IR light passes through the crystal and enters the first couple micrometers of the sample. This technique is the most versatile of the three and can be used to analyze a wide range of samples from electronics to cloth, all without sample preparation. So that's a short overview of FTIR spectroscopy. If you want to know more, check out our basic series here on YouTube, where we go into more detail. Now, FTIR spectroscopy lets us analyze a wide variety of materials, making this technique applicable to virtually any industry or field of study. But if FTIR spectroscopy can analyze all these different kinds of samples and materials, why do we need FTIR microscopy? Well, say we wanted to analyze the craters on this piece of coated metal to see what might have caused this defect. Each crater is quite small, maybe only a few millimeters across, and to figure out what's going on here, we need to selectively analyze one of the craters. When we make FTIR measurements on the spectrometer, we don't have the precision to choose where on a sample we want to analyze, so it's not a good technique for analyzing the craters on this piece of metal. But with FTIR microscopy, we can select the exact spot we want to analyze. FTIR microscopy can still analyze that same broad range of sample types, but now we have a lot of precision with our measurements. FTIR microscopes have so much precision that they can measure structures down to the micrometer scale, which is around the size of bacteria. A really common use of FTIR microscopy is failure analysis, like analyzing this piece of wire that has a defect in the plastic sleeve. We'll use the FTIR microscope to find the root cause of this defect. Let's see what goes on inside the microscope while we do that. To do this analysis, we'll need two light sources: an infrared light source to do the chemical analysis, and a visible light source so we can see and visually examine our sample under the microscope. Now, microscope optics are traditionally made of glass, but many frequencies of IR light can't travel through glass like visible light can. So what do we use instead of glass? Well, one solution is to use an IR transparent material like germanium for all the microscope components so the IR light can pass through. While this approach seems pretty straightforward, there's a couple complications. First of all, not all the IR light actually goes through these IR transparent materials. So as the IR light travels through all the components of the microscope, we lose some light here and there. By the time the IR light reaches the IR detector, we've lost quite a bit of IR light, which can make it more difficult to create a high-quality spectrum. Secondly, some of these IR transparent materials aren't transparent to visible light, So we'd end up with the opposite problem we had with glass. The IR light could travel through the microscope to do the chemical analysis, but we wouldn't be able to visually examine our sample. Finally, components made out of IR transparent materials tend to be very expensive. For all these reasons, the most common approach is to use a series of mirrors made out of a metal with high reflectivity for IR light, like aluminum or gold. By using mirrors, we can easily direct both the IR light and visible light around the microscope. We can even make the microscope objective out of these metal mirrors to focus the light on the sample. These special objectives are called Cassegrain objectives. So mirrors aside, how do we analyze the defect on this piece of wire? First, we'll need to do a little sample preparation. We usually don't need to do any sample prep for FTIR microscopy, but in this case we want to analyze underneath the plastic sleeve, so we'll hold the wire in place and cut the plastic away. Now we can see the defect, but this area is pretty small, about 2 millimeters long and 1.5 millimeters wide. So to get a better look, we'll place this under the microscope. First, we want to see our sample, so we'll direct visible light with the mirrors through the objective to the sample. The visible light travels back through the objective to either an eyepiece or a camera. Most IR microscopes just have a camera because cameras let us view the sample on a computer screen and take pictures. But the traditional microscope eyepiece still has its place in certain applications. Now that we can see our sample under the microscope, it's time to do the FTIR analysis. This lighter region on the sample corresponds to some contamination, so we'll select a point in this region to analyze. Our sample needs to interact with the IR light for the FTIR analysis, so the IR light will now travel through the microscope to the sample in a similar way that the visible light did. The exact way the IR light travels through the microscope here depends on the measurement technique we use, that is, transmission, reflection, or ATR. For this sample, we'll use ATR because it offers very high spectral quality. Now the IR light interacts with the sample, and then we'll collect the light and guide it towards the detector, which will register the IR light so we can obtain our FTIR spectrum. But there's one issue. We only want to analyze and get the FTIR spectrum of this specific point on the sample. The objective did focus the IR light on this point, but the IR beam is still larger than the region we wanted to analyze. So if we detected this IR light as it is right now, we wouldn't end up with a clear picture of what's happening just in our region of interest. So what do? We use an aperture. Apertures block some of this IR light from reaching the detector. That way, we only detect the IR light from the point we want to analyze. There's a couple different aperture styles we can use in our microscope. The simplest kind of aperture is called a pinhole aperture, which is just a small hole that's close in size to the region we're analyzing. There's usually a wheel of different sized apertures, so you can select a size that's pretty close to what you need. The more advanced option is a knife edge aperture. Knife edge apertures are made of 4 blades. These blades can move to make a rectangular shape of any size in any orientation. That way, this style of aperture can exactly match the size of the region on the sample we want to analyze. So we send the IR light through an aperture to block the extra light from reaching the detector, and now the IR light is detected and we'll get our FTIR spectrum. To figure out what the contamination on the wire is, we'll use a reference library. The reference library contains thousands of spectra of different molecules and compounds, and we'll search through it until we find a match. To make things easier, computer software can search through the reference library for us to automatically identify our sample. A reference library search on this spectrum identified the contamination as a mixture of PVC and fluorocarbon. Now that we know that, we can figure out what caused the defect on this piece of wire. But there's one more part of this microscope we haven't really discussed: the detector. The detector collects IR light so we can get the FTIR spectrum. To get high-quality results, the detector has to capture enough IR light. But often with FTIR microscopy, we're analyzing very small areas with a very small aperture, so a lot of IR light is blocked from reaching the detector. That means if we want to analyze really small areas, we'll need a really sensitive detector to get an accurate analysis. Luckily, there's several detector options available for FTIR microscopes, so we can get the sensitivity we need. Going from least sensitive to most sensitive, we have DLA-TGS detectors, TE-MCT detectors, and LN-MCT detectors. With the most sensitive detector, the LN-MCT detector, we can analyze samples as small as 5 micrometers, which is about the size of a human red blood cell. However, this super sensitivity comes with a trade-off. These are high-maintenance detectors that must be continuously filled with liquid nitrogen for cooling. TE-MCT detectors have slightly lower sensitivity. They can measure anything larger than about 10 micrometers, which is about the size of a flour particle after milling. However, they are continuously cooled with thermoelectric cooling, so they're more convenient to use. DLA-TGS detectors require no cooling at all, but they can only measure samples larger than 50 micrometers, which is about the size of the average human hair. That's still small, but it's in the range of things we can already see with the naked eye. So DLA-TGS detectors are limited to applications where we're analyzing areas that are relatively large, in the range of what we can see without a microscope. Now, all these detectors are called single-element detectors. Single-element detectors make one FTIR measurement on one point of the sample at a time, so you end up with one FTIR spectrum. There's a whole other class of detectors called imaging detectors that can measure over 1,000 points on a sample at once, so you get much more IR data. Another difference between single-element detectors and imaging detectors is that imaging detectors don't require an aperture, so all the IR light is captured during a measurement, leading to faster and higher quality results. We need the capabilities of an imaging detector to create IR images, which are colored images that help us visualize the chemical composition of a sample. IR images are made of thousands or even millions of FTIR spectra. But IR imaging and imaging detectors are a topic deserving of their own video, so we'll leave this discussion here. Now we know how an FTIR microscope works, so let's quickly touch on a few applications. FTIR microscopy is useful when we want to analyze small structures or samples, or specific areas on a sample. That makes it convenient for failure analysis and quality control to quickly identify contaminants or defects in products. In forensics, it can analyze paint layers or small fibers that may be evidence. And in environmental research, it can analyze soil or small microplastic particles. An FTIR microscope can analyze lots of different materials, so there's many more applications of this technique. But for now, this should give you a good idea of what FTIR microscopy can do. Thanks for watching this video. We'll see you in the next one.